Hello, everyone. We're going to go ahead and get started. And if I could ask my friends in the back row to come on up, I do not bite. It'll make it easier if we have time for questions at the end for me to get that mic to you. But everyone, welcome to our 2024 conference. Right now, you're in our general track 1115 session, Immune System Basics and the Immune Dysregulation of PI Disorders. My name is Megan Messick, and I'm the Director of Education here at the Immune Deficiency Foundation. And I'm just so happy to be your moderator for this presentation. This session will be 45 minutes in length, and as I mentioned, we do hope to have time for questions if we can at the end of the presentation. I'd also like to share just a couple of disclaimers before I introduce our prestige guest. The first is that the information in today's session is not intended to be medical advice. Always seek that information from your own health team and healthcare providers. Additionally, here at IDF, we offer a wide variety of educational programming, including those provided by life management professionals and others in our healthcare community. The views and opinions expressed by our guests today do not necessarily reflect the viewpoints here at IDF. But without further ado, I am so honored to introduce Dr. Christopher Chang. Dr. Chang is Chief of Immunology, Allergy, and Rheumatology at Joe DiMaggio Children's Hospital and Memorial Healthcare System. He currently serves as the Program Director of the Allergy Immunology Fellowship Program and Associate Program Director of the Transitional Year Residency Program at MHS. For full presenter bio, you can continue to read more about our guests and during your conference application review. Dr. Chang?
Thank you, Megan. So I'm aware that I'm standing between everybody and lunch, so so today I wanted to talk a little bit about the immunodysregulation, and this is actually a perfect segue, I think, because the last speaker had started to talk about non-infectious complications of immunodeficiencies. And so that's kind of what I'm going to talk a little bit about. And so I don't have any financial or other conflicts of interest. Today's objectives are to increase awareness of a new paradigm in immunodeficiency. And the previous speaker, Dr. Satter, actually alluded to this already. And it's not entirely that new, but it is gathering momentum as time goes on. And to understand the complex nature of the immune system, sort of on a conceptual basis. And we're going to be talking a lot about different aspects of the immune system, a lot of cytokines, but I don't want you guys to get kind of lost in the weeds of all the minutiae of cytokines. So more understanding on a conceptual basis why we have this new paradigm. And to raise awareness of associated clinical symptoms and signs and comorbidities in primary immunodeficiencies, because that's what this is kind of about, is the immunodysregulation in immunodeficiencies. And then again, cytokines, you may have heard the term before. They are actually very important molecules, and I wanted to kind of emphasize their role in health and disease. So this is kind of how our evolution, the evolution of our knowledge of immunity. Obviously, we've known about the immune system since the late or mid to late 1800s, and it wasn't until the 1950s that we sort of discovered that maybe these things were genetically determined. In the 1970s, we had the story of the boy in the bubble, and in the 1980s to the 2000s, we started understanding mechanisms of immune regulation. And then cytokines, which the first one was discovered actually in the 1950s, but the term cytokine was actually not coined until the 1970s. Incidentally, that first cytokine that was discovered in the 1950s was interferon alpha. In the 2000s, there was a rapid increase in our discovery of monogenic immunodeficiencies. This contributed to this molecular diagnosis of primary immunodeficiencies, where a lot of your common variable immunodeficiencies, CVIDs, now actually have a genetic cause for them and are able to be more accurately defined. And so this began the period of these inborn errors of immunity that we talk about now, that as a term that in many cases has replaced immunodeficiencies, and the role of immunogenetics, which is really important. And as Megan mentioned, I'm the program director for an allergy immunology fellowship program, and immunogenetics has become a big part of the education for our fellows. And then in the 2010s, we recognized the relationship between immunodeficiencies and autoimmune diseases, and this sort of got expanded into, it's not really just autoimmune diseases, it's actually other types of inflammatory disorders that maybe could caused by viruses and things like that. And we saw that in COVID-19 with these cytokine release syndromes and cytokine storms, cancer and immune regulation, and the newest probably thing that has come about is the coining of the term primary immune regulatory disorders, or PIDDs, and there are several forms of this. So immune dysregulation and primary immunodeficiency can take many forms. Autoimmune diseases, we've known for a long time that people with primary immunodeficiencies also can have autoimmune diseases, and at first glance, this seems to be a paradox. How can someone with supposedly an insufficient amount of immunity, enough to cause immunodeficiency, also have too much immunity that can cause autoimmune diseases? And we're gradually more and more understanding why this is. Gastrointestinal disorders have always been sort of part of immunodeficiencies, inflammatory bowel diseases, and so forth. The lymphoproliferation, we're discovering that there are lymphoproliferation syndromes like GLILD that we see in CVID and Epstein-Barr virus-associated lymphoproliferative symptoms in diseases like XLP. And then even allergic disorders has in some way or form is related or associated with primary immunodeficiencies. There are lots of diseases like hyper-IgE syndrome, hypereosinophilia syndrome, STAT gain-of-function mutations that produce an immunodeficiency but also produce an allergic disease. And the International Union of Immunologic Societies has categorized inborn errors of immunity into 9 main categories. And you can see down there, number 4 is diseases of immune regulation. These are the primary immune regulation disorders, the PIRDs. And the PIRDs consist of hemophagocytic lymphohistiocytosis, or HLH. This has been associated with many primary immunodeficiencies and can occur in both primary and secondary forms. Then you have syndromes with autoimmunity and others. And so this is a categorization that they've come up with. So they have ALPS in this group, which are disorders of apoptosis, syndromes with regulatory T-cell defects like IPEX and LRBA, and syndromes without regulatory T-cell defects like the APOSED and the autoimmune polyendocrine syndromes. And then you have immune dysregulation with colitis, such as the IL-10R deficiencies. And the PIRDs, primary immune regulatory disorders, there are many of them now, but they overlap with CVID, IPEX, and CTLA4, LRBA. These are all genes that have been discovered now that are sort of part of this inborn errors of immunity. What's important to realize is that people with immune regulatory disorders and an immunodeficiency don't always present first with an immunodeficiency. They can present with other clinical manifestations before we even find out they have an immunodeficiency, such as autoimmunity or GI symptoms or autoinflammatory signs or symptoms like fever and rash and so forth, and signs of acute or chronic inflammation. In HLH in particular, there are, as I mentioned, primary HLH and secondary HLH, and the primary HLH are often genetically determined, and so you have variants in the genes that are associated with HLH, such as STX or syntaxin 11, PRF1, which codes for perforin. These are kind of those specific genes that have been known to cause primary HLH. But immunodeficiencies can also present with HLH, such as in CD27 deficiency or Chidia-Kagashi syndrome, Griscelli syndrome, Hermansky-Pudiak syndrome, ITK, and interleukin-2 inducible T-cell kinase deficiency, X-linked proliferative disease, XLP, and X-men, which is a disease caused by defects in MAGT1. In secondary HLH, there are multiple causes. Infections can be a cause. Autoimmune diseases can be a cause. In fact, the rheumatologists have their own special name for HLH when it occurs in their JIA patients. They call it macrophage activation syndrome. It can be triggered by infections, EBV, even COVID, bacterial, parasitic, fungal infections. And this is probably one of the reasons why we see it in our immune deficiency patients, is because they are susceptible to infections, and then the infections can drive a host response that can manifest as HLH. Other potential causes of HLH are solid organ or stem cell transplantation, can be iatrogenic immunosuppression, metabolic, or even trauma. So the cardinal signs of inflammation, and this is very basic, the signs of inflammatory response are heat, pain, redness, swelling, and loss of function, right? Calor, dolor, rubor, tumor, and functio liesa. So the heat part is usually fever, manifests as fever, or warmth. Pain can be pain, but it can also be itching or other sensations of the skin. Redness is really referring to like a rash or flushing or maybe color changes, and then the tumor can be swelling or angioedema. And then that last one, loss of function, could be many things. It can be organ dysfunction, it could be decreased mobility, but these are kind of signs of inflammation. So if you have some of these signs, that could indicate that you actually have something related to immune dysregulation. So our key players in immune homeostasis, and by immune homeostasis, really I mean good immune health. And the immune system is key to your health, right? And we're finding that more and more, that immunology is key to almost every other medical discipline. You have to know some immunology, and no matter what field you end up practicing, right? So these immune cells and factors really determine how healthy you are. In the end, it pretty much boils down to this, among other things, but these are really important players. So for T cells, there are T helper cells, there are T cytotoxic cells. Regulatory T cells, very, very important, right? Because if you're suppressing T cells globally, like all T cells are being suppressed, you might have an immunodeficiency. But if you're also suppressing T regulatory cells, what might you have? You might have a deficiency in the ability to regulate your immune response, and this could be one of the mechanisms for why you can have immunodeficiencies occurring at the same time as having an autoimmune disease. Macrophages are part of the innate immune system, and they are activated by T cells, and they play a big role in inflammation, right? And cytokines, there are different types of cytokines. There are interleukins, interferons, growth factors, right? And these cytokines can either be pro-inflammatory or anti-inflammatory, but many of them are both pro-inflammatory or anti-inflammatory, so it gets even more complicated. And then from the cytokines, you have these signaling pathways, and you hear about all these new medications now called the JAK inhibitors, right? And these are because the JAK inhibitors are the signaling pathway from the level of cytokines interacting with the receptors all the way down to STAT proteins and then the nucleus. And I'll show you a picture of this in a minute. And then you have your transcription factors, which drive the protein synthesis and turning on and turning off of genes, right? So cytokines, a key part of the immune regulation, derived from a combination of two words, cyto meaning cell and kine meaning movement, right? And so these are short-lived proteins, and they're released by one cell and helps to regulate the function of other cells, including potentially itself, and they thereby serve as intercellular chemical messengers. They are immunomodulators, and anomalies in cytokine function can lead to disease or death. So cytokines really play a big role in health and disease. What's the difference between cytokines and hormones? Well, they both kind of stimulate function in your body, but cytokines are generally present in much lower concentrations in the body. Having said that, when you activate a cytokine, you can, the activation leads to sometimes several-fold increase in the levels, whereas in hormones don't usually increase that much. Right? So cytokine activation compared to growth factors, for example, growth factors are usually constitutively produced. That means they're there all the time, whereas cytokines, sometimes you have to stimulate them in order for them to become activated. Now, cytokines can actually signal through several mechanisms. We call it autocrine, paracrine, or endocrine. Autocrine means it acts on itself. So a cell releasing a cytokine acts back on itself. Paracrine means it acts on something adjacent to it or nearby. And endocrine, acts on something far away in the body. Right? So it can act by any of these three mechanisms. And as I mentioned earlier, interferon alpha was the first cytokine to be discovered. And it was discovered as an antiviral soluble factor. Right? So you could actually transfer that factor, and it would suppress viral replication. And now we know that cytokines can also be membrane-bound. The cloning of the first interferon occurred in 1980s, and the first monoclonal antibody to a cytokine was actually against CD3. It was anti-OKT3. And it can work in 4 ways. Cytokines are very versatile. Right? They can work on their own, or there's pleiotropy. In other words, they can act in different ways depending on the cell they're targeting. Or they can work 2 or more cytokines and work together to create an effect on a target cell. That's called synergism. They can be they can produce an anti-effect, so they can antagonize, and so they can stop something from happening. And in the immune system, one of the problems with trying to understand the immune system is that there's so much redundancy in the immune system. And we see this with a lot of the studies on the monoclonal antibodies that we're trying to develop to target certain cytokines. You may be able to target one cytokine, but the immune system's so redundant that that pathway can still keep going based on another different cytokine. So these are the kind of the 4 ways cytokines can work. Cytokines are classified by function. So, type 1 cytokines are considered the ones that have effect on cellular immunity, type 2 cytokines on antibody production, or they can be classified based on their structure, their molecular structure, and you can kind of group these together, and these are how you have groups like the IL-1 family, IL-17 family, or you can group them by their receptor. Now, every cytokine, in order to exert its function, has to have a receptor. So it has to bind to a receptor, and once you bind to a receptor, then you can initiate that downstream signaling pathway. But there are different receptors, like there's the immunoglobulin superfamily, named because a lot of these receptors have structures that are similar to antibodies. Growth factor receptors, the interferon receptor family, is a family of its own. A huge family, the TNF, tumor necrosis factor superfamily, includes also non-cytokine ligands. And then another form of cytokines called chemokines, and chemokines are basically another separate class of cytokines, generally smaller molecules than cytokines, that help direct cells where to go. I guess that's why they're called chemokines. And then these are considered these G protein coupled receptors. So cytokines are produced by cells as part of normal cellular activity or as a result of an environmental trigger. They bind to receptors, they trigger these signal transduction pathways that I mentioned, and they initiate synthesis of new proteins. Now, when a cytokine is activated, the concentrations may increase several-fold. They're proteins, they're low molecular weight, they bind to the receptor and triggers a signal. They have functions in cell growth, cell differentiation, cell death, and apoptosis, which is a term that just really means programmed cell death. It means that the cell actually plans on how it dies, and this apoptosis is really important because it's thought that if you don't have that controlled, programmed way of cell death, then you release a lot of debris into the circulation, and that can trigger autoimmune diseases because now you have all these proteins that were released uncontrolled that can initiate an autoimmune response. Cytokines also can induce non-responsiveness or responsiveness to other cytokines, and then they can also induce secretion of other cytokines. This is just a timeline of HLH, and which was first described in 1952, but cytokine storm then this term was introduced, and this was introduced because of the cytokine release resulting from CAR T therapy in graft versus host disease. We developed guidelines and criteria for the diagnosis and treatment of HLH in 1994. They were revised in 2004. At this time, they're considered maybe to be not quite as useful because now we have a better understanding of the different types of cytokine release syndromes. Cytokine release syndrome was actually described in the first SARS pandemic or epidemic in 2003 and 4, and then of course cytokine storm in SARS-CoV-2. That really is what brought a lot of our attention to the role of cytokines in viral infections, and certainly can occur not just in SARS-CoV-2, not just in COVID, but also in other viral infections like influenza or metapneumovirus and so forth. So this is just a diagram showing you on the left. Do I have a pointer? Oh, pointer. No, okay. So on the left of the slide, you see the cytokine, and it binds to a cytokine receptor on the membrane, and that triggers the signaling pathway, the JAK-STAT signaling pathway. You can see JAKs and STAT3 and 5. The STAT then dimerizes and then goes into the nucleus and affects gene regulation, protein synthesis, and cell proliferation. So this is kind of how that pathway works. And I kind of wanted to mention that because when you have monoclonal antibodies developed to a particular cytokine, that is very specific. It really works on that cytokine that it's designed for. But JAK-STAT drugs, you hear a lot of the new JAK-STAT pathways, which actually were developed after cytokines, are much less specific because a lot of the cytokines use the same JAK-STAT pathway in order to induce gene regulation and protein synthesis. So in order to understand cytokines, you also have to understand that there are cytokine receptors. And cytokine receptors are also categorized. So there are cytokine receptors that contain the common gamma chain. And so IL stands for interleukin, which is a huge class of cytokines. And you have interleukin-2, which drives T cells, T cell development, T cell proliferation. IL-4, which is a sort of an allergy type cytokine. IL-7 is another T cell cytokine. IL-15, also T cell and NK cell cytokine. And you can see that they all share a subunit in their receptor. So if you're missing that subunit, you can see that can affect a lot of different cytokines, and that can affect a lot of different processes, disease processes. So there's another group that share the common beta chain, like GM-CSF, which is a growth factor, IL-5 and IL-3. IL-5 is your eosinophil cytokine, so IL-5 drives eosinophils to proliferate and be activated, and IL-3 drives basophils. And then there's a whole bunch of other shared cytokine receptors like GP130 or IL-4 receptor alpha, IL-10 receptor B, IL-17 receptor alpha. And so the cytokine receptors are often defined by their common shared subunit. Classes of cytokines, your interferons are a big class of cytokines. Then we talked about your interleukin family, pro-inflammatory cytokines, and the IL-1 family of pro-inflammatory cytokines includes IL-1, IL-18, IL-33, and IL-36. There are other pro-inflammatory cytokines like IL-2, and interleukin-6 came to many people's attention because of the use of tocilizumab during the COVID pandemic. It was really trying to block that pro-inflammatory or that inflammatory cascade, and I don't think it worked very well in COVID-19, but that's kind of the idea where that came from. And then tumor necrosis factor alpha is also a pro-inflammatory cytokine. And then you have the anti-inflammatory cytokines like IL-10 or TGF-beta. we'll talk a little bit about the individual cytokines coming up next. So the other thing that's important in the whole inflammatory process is that in addition to having the cytokines, there's something called an inflammasome. And inflammasomes are multi-protein complexes that lead to the production of IL-1 beta and IL-18. And so they're in the cell, in the cytoplasm of the cell, and when they're activated, they are assembled, and then they produce IL-1 beta and IL-18, which, as you heard, are really potent pro-inflammatory cytokines. And this is just a picture of the inflammasome. It has a lot of different effects in inflammation. Activates the inflammatory response, mediates apoptosis, is a part of your innate immune system. And there are different inflammasomes based on the sensor which drives the assembly of that inflammasome. So if the sensor is an NLR, a nod-like receptor, then they name it after that. And NLRs are these they're similar to toll-like receptors, so they're pattern recognition receptors and part of the innate immune system. This is the IL-1 superfamily, and you can see that there's several members of this family, and in general they're mostly pro-inflammatory, but you can see that some of them are actually anti-inflammatory. So this gets even more complex when you talk about how these cytokines are grouped together. And all of these cytokines, most of the cytokines in this family are on chromosome 2. So, they kind of group together also on where their genes are located. IL-6, again, we talked a little bit about that. Tocilizumab is a monoclonal antibody against the receptor for IL-6. There are other IL-6 inhibitors, like siltuximab or sarilumab, and satralizumab. And then these are secreted by macrophages in response to PAMPs. And PAMPs are pattern-associated molecular patterns. They are part of the immune system. It's the reason why the innate immune system has a faster response, because they recognize patterns and they don't have to actually be that specific and develop recognition to individual antigens through antibodies and T-cell receptors. So this is part of the innate immune system, works faster, and can be very effective, actually. But this is IL-6. IL-17 has a role in autoimmunity. It's expressed by T helper 17 cells, and it promotes autoimmune diseases. And there are several forms of the receptor. The signaling, it does help protect against pathogens. So you can see, again, it has a dual function. It protects against pathogens, but also promotes autoimmune diseases. So we need it, but too much is going to drive production of autoimmune diseases. Activates signaling pathways through NF-kappa B, and there are multiple targets now for IL-17. Brodalumab, ixekizumab, secukinumab, and they're all approved for the treatment of an autoimmune disease called psoriasis. So IL-17 is important in autoimmune disease because it drives autoimmune disease, but T regulatory cells are supposed to modulate that response. And so if you don't have enough T regulatory cells, or your regulatory cell function is not enough, then you're going to tend to develop more autoimmune disease, autoinflammatory diseases. Basically, your immune system will go unchecked, right? And so there's this balance between T regulatory cells and Th17, which is one of the hypotheses for the development of autoimmune disease. If your Th17 activity is too high, then you'll get autoimmune diseases, right? And if your Th17 activity is too low, then you may not be able to fight off certain types of infections. IL-22. So IL-22 is a member of the IL-10 family. IL-10 family of cytokines are thought to be anti-inflammatory. So when we see patients with these cytokine storm or HLH or these inflammatory or autoinflammatory diseases, and we look at their cytokine levels, we'll see that many times their pro-inflammatory cytokines are elevated, like the IL-1 beta, IL-18, interferon gamma, TNF alpha. These are all pro-inflammatory cytokines, and their levels are high. But what's interesting is that IL-10 levels are also high. And I think that's the body's kind of compensatory response, the feedback loop, that if you have too much pro-inflammatory cytokines, then you're going to also produce anti-inflammatory cytokines to counter that. And so you can see that IL-10, IL-22, IL-26, 28, 19, 20, these are all members of the IL-10 family, and many of them share a similar receptor subunits. And again, they mediate all through the JAK-STAT pathway, which is kind of interesting because it's actually a very non-specific pathway. IL-23 is a pro-inflammatory cytokine that shares subunit with IL-12, and IL-12 and IL-23 play a big role in autoimmune diseases like psoriasis. In fact, there are monoclonal antibodies against the IL-12, IL-23 common subunit that are used in the treatment of psoriasis, like ustekinumab or Stelara, and so this also has a role in autoimmunity. Tumor necrosis factor is a huge group of very important pro-inflammatory cytokines, and it is a member of the tumor necrosis factor superfamily, which includes a lot of other molecules which are not cytokines, and these classifications are based on their common sequences. The receptor for TNF is also a huge superfamily, so there are probably TNF has 19 ligands that interact with 29 receptors, so you can imagine how complicated this can get. And then the TNF receptor 1 is a 55 kilodalton receptor, and the receptor 2 is a 75 kilodalton receptor. This is a huge family because tumor necrosis factor is evolutionary conserved and can be tracked back to very primitive organisms, and they regulate cell differentiation, survival, and apoptosis, and they control a lot of the innate and adaptive immune cells. They're mostly pro-inflammatory, and again, they signal through the same signaling pathways we talked about earlier, NF-kappa B, and they can also trigger apoptosis and cell death. So tumor necrosis factor is another very important cytokine. This slide is a little bit out of place. We talked about it already. But IL-10 receptor is shared also between, as I mentioned, between all the other members of the IL-10 family. So I've talked a lot about these monoclonal antibodies already, and all this is possible because of the discovery of hybridoma technology, which is the ability to make large amounts of a single antibody, a monoclonal antibody. Most antibodies that you produce in your body naturally are polyclonal. They're not monoclonal. But if you can produce a monoclonal antibody to target a specific antigen or protein and make large amounts of it, you can use it to treat diseases. And so this whole experiment of hybridoma technology was discovered by Milstein and Kohler back in 1975. And we have now numerous monoclonal antibodies against different cytokines, actually. And this is some of them: IL-2, IL-3, IL-4, IL-5, 6, 8, 10. Some are not currently used yet. Like we don't have a monoclonal antibody against IL-10 as far as I know, or against TGF-beta. But a lot of these monoclonal antibodies have been produced targeting these cytokines. So I think it helps to know the mechanism, which is sometimes easier said than done because the mechanism is often very complicated. And to try to sort that out, I think when the golden age of immunology is probably after I finished my fellowship and that explosion of knowledge that we have now. Certainly, most of what I know now in immunology, I didn't learn as a fellow when I was a fellow back in the 90s because it just didn't exist yet. And now there's this explosion of knowledge, but I still think we've just basically touched the surface of the iceberg, the tip of the iceberg, that there's still so much more that we can learn. It's important to realize that immune systems have lots of redundancies, which kind of presents a little bit of a dilemma in how to choose treatments. Do you want to choose something that's very specific, or do you want to choose something that has broader effects? So the story about how steroids, which are used in the treatment of a lot of these comorbidities of immunodeficiencies, came about. It's an interesting story back that started back in the 1930s when they were trying to extract chemicals from adrenal glands, bovine adrenal glands, or cow adrenal glands, and to see which one worked, which one had biological effects. And they were able to isolate 6 different fractions from adrenal glands. Then the Second World War broke out, and the urban legend is that the Allies saw that the Luftwaffe could fly, pilots would fly at high altitudes without oxygen, and they wondered how. And they discovered that they were getting injections of adrenal extracts from cows. And then the result of that was they poured all this money into research on adrenal extracts, and after the war, they isolated one of the extracts was called compound E. They actually isolated 6 different compounds, A through F, but E was the one that they thought had the most biological effects. And they gave it to a patient with rheumatoid arthritis, and she just recovered. She was debilitated. They gave her the compound E, and she got up and went shopping, checked herself out of the hospital, you know, and that. But compound E was later found to be cortisone, and it basically was the first steroid. And the discoverers of this won the Nobel Prize 3 years later, which is almost unheard of. Usually they wait 30 years before they give you a Nobel Prize, but in this case it was very quick. And it's only after that we discovered all the side effects of steroids, right? All the bad side effects of steroids. And so this is one of the problems with steroids. Work very broadly. They basically suppress everything. I mean, you know, my childhood doctor used to give all his patients steroids. Everyone who came in with a cold and fever got steroids, and the fever went away. And you know, and so the problem is that when you have these broad-based treatments, they have a lot of side effects. The way I look at cytokines is this is much more specific, right? You're targeting a specific cytokine, but in doing that, you may omit some of the redundancies of the immune system, and they may not be as effective. But then you get to JAK inhibitors, which were discovered after cytokines, and we've kind of gone back to this broad-based treatment because JAK inhibitors will suppress that JAK-STAT pathway for any cytokine, almost any cytokine. And it's kind of like JAK inhibitors are like the steroids of the modern world. But you know, managing immune dysregulation is really a multidisciplinary approach. This is just what I talked about with the targeted treatments and the broad-based immunosuppressive therapies. But it does require a multidisciplinary approach, and at our hospital, we actually formed an immunodysregulation response team, and it involves the cooperation of all these specialties: rheumatology, hematology, oncology, the hospitalists, the intensivists, the neurologists even, you know, infectious disease, which I guess I neglected to put in there. And they all come together and we all treat these patients as a group. When we developed it, you know, we had to get everybody together, we had to agree on an approach. We selected immunology, in our case, as a primary service, but it could be any of these disciplines. We developed a clinical pathway, order sets, implementation, and we're constantly evaluating our outcomes and improving our approach to this.